In this concept video, you will be learning about the chemical structure of nucleic acids. The monomers of nucleic acids are nucleotides, and nucleotides are basically nucleoside phosphates. The addition of a nitrogenous base to a pentose sugar forms the nucleoside. The further addition of the phosphate group forms the nucleotide. The addition of the phosphate group to the fifth carbon of the pentose sugar forms a phosphodiester bond. This slide shows how the nucleic acid polymer forms through the formation of further phosphodiester linkage between the preceding nucleotide and the third carbon of the next nucleotide. This reaction is a condensation reaction, as a water molecule is removed in the process. Thus, the formation of phosphodiester bonds is through condensation reactions. When many phosphodiester bonds form between the phosphate group and the third or 3' and fifth or 5' carbon atoms of the pentose sugars, an unbranched sugar-phosphate backbone results. The nucleic acid has end-to-end chemical orientation, which in this case shows the 5' end to 3' end from top to bottom of the nucleic acid. Nucleic acids are linear polymers of nucleotides. There are two classes of nucleic acids, namely deoxyribonucleic acid (DNA) and ribonucleic acid (RNA). Note that RNA has uracil in place of thymine as one of the 4 bases it contains when compared to a DNA strand. DNA consists of 2 chains of polynucleotides which are coiled around each other to form a double helix. These 2 chains are held together by weak hydrogen bonds formed between nitrogenous bases on the opposite strands. This base pairing is complementary, with hydrogen bonds formed between A and T as well as between C and G. The 2 strands are also antiparallel. One strand is oriented in the 5' to 3' direction, while the other is oriented in the 3' to 5' direction.